Okay, everybody ready? I'm going to assume that is a yes. Somebody give me a yes.
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 Great. Okay, so welcome to another Aspirational Neuroscience Journal Club. This time we are covering this paper entitled Fear Learning Induces Synaptic Potentiation Between Engrams Neurons in the Rat Lateral Amygdala by Abatis. It was published in Nature Neuroscience in 2024. The authors used a very technically challenging technique, simultaneous 12-cell patch clamping, to determine the functional connectivity between lateral amygdala fear memory engram cells. And what they find is that fear conditioning specifically causes potentiation of synaptic connections between learning recruited neurons. These findings of synaptic plasticity in an auto-associative excitatory network of the lateral amygdala may suggest a basic principle through which a small number of pyramidal neurons could encode a large number of memories. I chose this paper because I think it reveals some general principles regarding how memories are formed. Namely that synaptic connections onto and between engram cells are strengthened, forming an engram ensemble that is both selective to and to a particular input stimulus, and that is effective in amplifying that stimulus to drive later behaviors. I also chose this because I think it suggests a possible connectomics experiment, which could demonstrate the decoding of a non-trivial memory from a static connectome. And I'll discuss this at the end of the talk, and I'm looking forward to feedback on whether this would actually be considered a non-trivial enough to, for example, win the Aspirational Neuroscience Memory Decoding Challenge, which is, of course, what these journal clubs should be kind of gearing toward. Okay, so before going on to the main paper's results, I'm going to review what I believe to be the main theory of how auditory fear memories are formed, to help put this paper's findings in context. This is probably dramatically oversimplified, but that's where we are. So first we have the auditory thalamus, the medial geniculate nucleus, and the auditory cortex, which I'll just summarize as auditory cortex for the rest of this talk, which represents distinct sounds by distinct patterns of activation over their neurons. And the axons from these auditory regions project to the lateral amygdala, as shown in this diagram. The lateral amygdala cells also receive input from pain circuits. I'm not showing that here, but they just receive input from pain circuits directly. And the lateral amygdala axons project to downstream areas, central amygdala and later, to instigate freezing behavior of the animal. The lateral amygdala cells also connect to each other recurrently, as shown here. These connections are the main focus of today's journal club paper. For the description of learning that I'm about to lay on you guys, I want you to assume that there's a lot of silent synapses in this box here. So silent synapses are synapses that have NMDA receptors so that they can sense coincident activation and undergo Hebbian strengthening, but they don't provide a significant signaling prior to learning. So just assume that there's some small synapses there. Now, during an auditory fear event, the sound produces a unique pattern of activity across the auditory cortex, as shown here, while simultaneously a random unique subset of lateral amygdala cells becomes active due to the pain signals. This subset of lateral amygdala cells is believed to be chosen at random due to the cell's intrinsic excitability, which fluctuates on a day-to-day basis. So it's a random subset. The previously silent auditory cortex to lateral amygdala synapses that I just showed there, that right there, undergo LTP and strengthen. And at the same time, according to today's journal club paper, these lateral amygdala recurrent synapses also strengthen, these here. And so to summarize this oversimplified picture, this is what is thought to be the neural substrate of a fear memory. Number one, an ensemble of engram cells in the lateral amygdala. Number two, synapses among these engram cells that endow the engram with autoassociative amplification powers. And number three, synapses from auditory cortex to the lateral amygdala that endows this engram with specificity for one sound in particular. And if that particular sound occurs again, let's say the next day, these synaptic pathways will amplify that sensory input signal and cause the mouse to freeze even without a pain signal. Now, there have been many papers that have shown the strengthening of these auditory cortex to lateral amygdala synapses that I'm highlighting here. But I think the best one that I found is this paper, this 2014 paper by Nabavi et al. And I'll take a few minutes to go through its results before I go through the main Abatis 2024 paper that really researches these recurrent synapses in the lateral amygdala. Everybody
Yeah, I was just going to ask you if you were able to hear when I raised my hand and if you want to be interrupted at all. Because
yes, go ahead and interrupt me.
So this refers to this slide, the previous slide, the one before. So I guess what I'm wondering is, and I also wondered while reading the paper, is what previous knowledge or models or theories are there about how it is that you choose, or that the amygdala can choose that this is actually something that should be remembered as something to be afraid of. Say, for example, achieving strengthening of the synapse in the presence of pain and sound, but only for pain that is above a certain level, or auditory stimulus plus the pain above a certain level, versus, say, just a very strong auditory stimulus or pain just by itself without any particular correlation to anything.
So the short answer is I don't know, but I've in the process of this, I read a review article on the lateral amygdala in general, and there are a bunch of modulating circuits also that go into this. There's dopamine circuits, and so I think what puts it over that threshold is probably just simply not in this paper and not in these diagrams.
Okay, yeah, this was I guess you said modulating circuits, so this is in line with another concern I had when reading the papers, that because of the way most of the studies were done, ex vivo, in vitro,
yes,
that a lot of the, say, rhythmic modulation and other factors that have to go into a proper description of the system, its modes and when it does something and how it switches, are not always included.
That's right. In fact, I touch on that later in the talk.
Okay, so, fantastic. Thanks.
I'll highlight that. I think there's a couple of things that that you're bringing up that I'll touch on later in the talk, actually.
Awesome.
Okay, so the Nabavi 2014 paper, what they did is they injected the auditory cortex and the auditory thalamus with an optogenetic virus to tag random subset of the cells. So that's what's shown here. This caused optogenetic channels to be expressed in the axons of these cells that terminate in the lateral amygdala. That way they could shine blue light with a little fiber optic cable onto just the lateral amygdala, and while the rat was being shocked. And this created a fear memory to this optogenetically activated fake sound conditioned stimulus. And presumably, it is strengthening these synapses. That's what I'm going to be talking about here. And so in that paper, here's the plot that they show that shows that the memory was stored. So the y-axis here is lever presses, they could see that the rat was freezing if it stopped a lever pressing task. And so when they shined the blue light, it froze. Now, where the authors really show that this memory is encoded in these particular synapses is here. They used a laser light, the same laser light, to perform a LTD, a long-term depression protocol. So that's when they slowly activate they have a slow 1 hertz action potentials that specifically shrink those synapses. And when they do that, behaviorally, the memory is erased. Then they could go and do a long-term potentiation experiment where they rapidly activate these auditory cortex axons to strengthen those synapses back up. And they show that, lo and behold, the memory comes back. And they can do that multiple times. And they have a quite robust result that they could erase and restore this behavioral memory simply by shrinking and regrowing these or re-expanding these synapses from the auditory cortex onto the lateral amygdala. So I wanted to make clear that there's a bunch of cool results that talk about those key synapses. But right now we're going to go to the main paper, which talks about these lateral amygdala to lateral amygdala synapses, and as shown here. So this Abatis et al. 2024 paper, I'm breaking it down into about 6 experiments. The titles they have, I've numbered them though. There's actually just one experiment here, this ex vivo connectivity after fear conditioning, that I think has the main clear results. So I'm going to spend most of my time on that. So in experiment 1, the authors determined the baseline connectivity statistics of the lateral amygdala. So what they did is they took a rat, chopped its head off, sliced a 400 micron thick slice out of its brain containing the lateral amygdala, put that in artificial cerebral spinal fluid so it was still alive, and did 12 pipette whole cell patch clamp recordings on cells in the lateral amygdala. As shown in this diagram, current was injected in one of the cells, and then they listened to the other so that it produced a train of action potentials while they were listening to the other 11 cells, monitoring them for micro excitatory postsynaptic potentials, which is going to happen whenever there's a glutamate vesicle that is released because of that action potential. So if they saw any of those, then that's a sign that the cell that they're driving is connected to the one, is synaptically connected to the one that they're listening to. They used 34 rats and total 90 slices to test a total of 637 excitatory neurons with 4,157 possible connections. So they listened to all of those pairs through all those experiments, of which they found 89 actual connections, which implied a 2.1% probability of synaptic connections between any 2 excitatory lateral amygdala cells. They did a whole bunch of other things that are really cool, but are probably not so relevant. They calculated the quantal release size. 460 microvolts per vesicle released. They calculated the 5 release sites on average per functional connection. Also, what they did is that is relevant to a potential connectomic study is they because they backfilled all these neurons, they knew where the cell bodies were, and they found that cells that are closer to each other, like within 100 microns, had a higher probability of connecting with each other. So it maxed out at about 3.5%. And so that was kind of a basic.
Can I ask a question about the previous experiment?
Please.
can you go back to it? I want to see the traces. So before these two images, yeah.
This one.
Okay, this one here, because there's something I didn't quite get, and that's probably just because I'm not an ephys person. So the red traces, obvious what's happening. Also, we should keep in mind that these here are the titanic pulses, where you're getting like a whole series of APs that are just not really giving you all the natural results that you would want to see for your EPSBs. But you're getting that on the next slide then, because that's where they do the single AP stimulation and show you what it is. But I'm wondering about the gray here. So the cell 1 to cell 2, these are patch clamp, right? So when you're doing extracellular, it's very normal that you're also stimulating a variety of cells that are nearby. But is this supposed to indicate that cell 2 is very strongly connected to cell 1, or is this an
I was wondering that myself. I don't know. Why are there off diagonals here? It's a great question. I don't know.
Okay, if there's anybody on the call who's an ephys person who does patch clamping, maybe you can chime in and clarify for us, because this is I was wondering about that.
Yeah. Sorry. And there's a whole bunch of other stuff that I'm not going to be able to answer on these electrophysiology experiments as well. So in experiment 2, they did more electrophysiology on the same slices. Basically, what they were doing is they were putting in action potentials on one, and now that they knew that there was a synaptic connection, they were trying to quantify the type of a synaptic connection, so they could classify it as a facilitating or stable or depressing synapse. From this, they calculated the overall temporal summation that could be expected at 183%. I'm not exactly sure what that means, but the main meaning is that if multiple input action potentials arrive simultaneously, then the synapses have are more effective. Okay, they did current injection experiments and showed that the average threshold for spiking, I think this is a delta, was 28 millivolts. Again, this is laying the groundwork for a statement that they're making later. So they put these together, and they estimated that an average of 34 of the synapses out of the total 230 synaptic inputs from lateral amygdala to lateral amygdala neurons for each neuron would have to be activated together to cause an action potential. And then they note that previous studies have shown that 10 to 30% of the excitatory neurons in the lateral amygdala are recruited into a fear memory engram. And they say this corresponds to a certain number of synapses. Actually, let me just read their thing here. Based upon previous findings, fear conditioning recruits 10 to 30% of lateral amygdala neurons. And on the premise that fear conditioning can indeed induce local synaptic potentiation that reaches a similar level of this temporal summation, this should allow for reliable signal propagation across the lateral amygdala. So what they're really setting up here is they're saying, look, from all the electrophysiology that we do, it makes sense that a signal isn't going to be propagated through if you don't have kind of a local ensemble that's going to increase the sensitivity, if you will. But if you do have that local ensemble, then and it's got lateral amygdala to lateral amygdala potentiated synapses, then it might very well amplify a signal down the line. Okay.
Yes, so that was actually one of the places where I was thinking, oh yeah, and this is where modulation is probably going to play a pretty big role as to whether you can achieve postsynaptic spiking or not. So those numbers, since they were done, I think that was in vitro. Yeah. So
in vitro. These are almost everything here is in vitro. Yeah.
Okay. Yeah.
Yeah. Or ex vivo slice, I guess is what they call it. This actually, so I'm moving on to experiment 3. Remember, experiment 4 is the one that I consider to be the main one that has meat. So in experiment 3, the authors performed a set of simple Hebbian LTP experiments in these acute slices. So this makes sense that they would do this, right? They've got the slices there. They've got neurons that are connected that they found. They can give, I don't know, 100 hertz stimulus to one and see if it strengthens its synaptic connection. And they did find that in this, but it was a super weak result, at least if I'm reading it correctly. They say that if you look in this figure, the first spike, I can't show my mouse. Do you see my mouse?
Yes, we do.
Oh, you do. Okay, so this first spike here, this is the facilitation of the first spike. So if they gave this LTP protocol, then there's a little bit of a significant rise in connectivity, but all the other spikes, spikes 2 through 8 of a train, didn't show any. And this was only on accommodating synapses and stuff like that. So I feel like even though they got a result here and they showed LTP, it's super weak, and I think that's because this was a slice and they weren't squirting dopamine on it, or it wasn't it didn't have the right things to actually what would happen in the animal.
I was wondering, I didn't read it carefully enough to catch this, but maybe you did. So when they were testing this, did they repeatedly provide the stimulus, or did they only do it once, or what exactly were they measuring here as the this is in consequence of what?
So my understanding is what they did is it probably says here, we use a standard Hebbian association protocol to associate 15 times 10 presynaptic action potentials with a postsynaptically evoked action potential at a delay of 10 milliseconds.
And so the result was after 15 sequences of 10 APs in a sense, or am I reading that?
I think so. I think so.
I see. Okay, yeah, in that case, it does seem kind of weak. Yeah.
It does seem kind of weak. And they get much stronger results for the next one. So that happened in the animal. So I would just ignore this one. So this is this is experiment number 4. I think this is the main experiment here. So in experiment number 4, the authors injected rats with a virus to mark all the cells in red and in the lateral amygdala. And those cells that were highly active within the last 90 minutes would produce green fluorescent protein. So they would be marked yellow. And they did this by using a degradable form of green fluorescent protein that would degrade over an hour, I guess. So if a neuron is active, it's going to be producing green fluorescent protein, but that green fluorescent protein is going to be degraded. So they're only going to see they're only going to see these yellow neurons when they sacrifice the animal 90 minutes after having undergone something that would activate those neurons. Okay, so they used so the experiment that they did is they injected the virus. Then they had 3 conditions. They had a home cage condition, a paired condition, and an unpaired. On week 2, day 1, on day 1, essentially, they would either just leave the rat in the home cage, or they would, in the paired condition, they would give the rat a tone and then overlap that with a shock. And in the unpaired condition, they would give the rat a shock, and then they'd wait like 10 minutes and then have a tone. So the idea is that only in the paired condition should these be should the tone be associated with the shock. Then on day 2 they tested it, right? So they gave a tone, and the images up at the top here, H, U, and P, home, cage, unpaired, and paired. In the that is images of a slice 90 minutes after they tested with a tone. So only those neurons that were active when they gave a tone on the second day are going to be yellow. And so that's why only the paired condition has a bunch of yellow engram cells. So those are the engram cells. Okay. Now, why did they do that? They did this because they wanted to do electrophysiology. So they did electrophysiology on the slice, but they could tell whether they were in a yellow neuron or a red neuron. And so this is the key result of the paper here. And it's funny. It's the key result of the paper. So, of course, they put it in extended figure 7. So they found significantly more connections between engram cells. 7.2% of engram to engram cells, engram cell pairs were connected, versus only 1.4% of non-engram cells. Not only that, the connections between engram cells were much more strong. So, and it showed up not just on the first spike, but all the spikes. So this answers your question, Randall, about I think this is way stronger result of LTP. So they had a micro EPSP response of 1.67 millivolts versus 0.2 millivolts, 0.24 millivolts for non-engram connections. And they also, in the same experiment, they tried to increase. They did an LTP again on these cell pairs, and they use this to show that this really is LTP, because if you try to do LTP on something that has already done LTP, then you don't get any LTP. And that's what they show. They only get LTP in the ones that were non-engram connections. The engram to engram synapses are already LTP'd out. That makes sense? So let me, so since this is the most important one, I'm going to read this. This is how they put it. This suggests that through a Hebbian mechanism, fear conditioning leads to synaptic strengthening between recruited lateral amygdala neurons, and may thereby ensure, after fear conditioning, a strengthening of local synaptic connections that can promote reliable signal propagation across the lateral amygdala. All right, we're getting there. Any questions on that one? That was the kind of crucial one. Everybody still there?
That one was very clear. I should say, though, for me, it feels like because theory tends to run ahead of experiment, and models do a lot of things where we're already assuming what they're trying to show in detail here. It feels like, yeah, I don't know. I mean, it feels as if they're showing things where you go, duh, of course. But that's because I buy into like the standard model of how this works, or how memories work in a sense. Yeah.
Although I haven't seen a paper that actually goes to these recurrent connections in lateral amygdala before this one.
Yeah, no, that's what I mean. Like you actually doing that empirically and testing it in great detail by actually looking at the individual synaptic sites and stuff, comparing with patch clamping, that takes a lot of work. And so it's like experimental physics always lags behind theoretical physics, and it's the same here. Yeah.
Okay, so the last 2 experiments are pretty quick. So the previous experiment, one problem with the previous experiment is that it could mean that those connections were already there. Okay, maybe that ensemble of cells was already connected together, and that's one of the reasons why it became the engram in the first place.
Just for clarity, when you say the connections were already there, you mean strong connections, right?
Strong,
exactly. Right. Because not making new synapses here. Okay.
Absolutely. That's a good point. Good point. So it could be that there was already really strong connections, and there didn't have to be LTP between these cells in order to strengthen those. They've just kind of picked out a strongly connected subset. So in experiment 5, what they did is they tried to address that. They used 32 fine wire electrodes mounted on little micro drives to do single unit recording in the living rat. And this allowed them to record neurons both before and after fear conditioning. So they could essentially retroactively ask the question, were these neurons that did become more connected, were they already more connected? Were they already at that level of functional connectivity before the fear condition? Now, unfortunately, of course, this means, because this is in vivo and they're using wire electrodes, they're not doing patch clamping. This means that they can only record spikes from neurons. So they can't do the type of direct electrophysiology that verifies that there actually a synaptic connection and to measure its strength. Instead, they had to rely on Granger causality, which my understanding is it's just a simple way of saying if neuron A spikes after neuron B, then you have some slight evidence that neuron B is.
That's right. And you don't know if it's a direct connection or an indirect connection through something else, all that. Yeah.
Exactly. So this is why this adds something, but it's not as good as experiment number 4. So in any case, what they did is they did the fear experiment, and they could see what neurons were spiking to say these are the ones that were recruited for the engram, and then they did Granger causality of the spike trains both before and after. And what they found is that there was an increase in the functional connectivity between engram cells, but only after the fear conditioning. And experiment 6 essentially does exactly the same thing, except using optogenetics to drive the recruited cells instead of just relying on their own firing. Okay, so to summarize the whole paper, this is the paragraph that I picked out. These findings reposition the lateral amygdala from a passive relay station into an active hub where synaptic plasticity strengthens lateral amygdala recurrent connections within neuronal ensembles following fear learning. Thus, fear memory encoding involves not only recruitment of intrinsically more excitable LA neurons and the potentiation of their external afferents, but also stronger binding of these neurons together in a local network that, after fear learning, can promote signal processing across the lateral amygdala. And that's all I have for the paper. The rest is a memory decoding experiment that I want to throw at you guys based upon this. So this is a good place for.
Sorry, Ken, to be the one who keeps on clicking the hand up button. So a thing
the hand up button. So
a thing that isn't in the paper, but that I was very curious about because I haven't studied amygdala in as much detail as hippocampus. You remember last paper, we had conversations about remote memory independent of hippocampus after a certain amount of time, hippocampus acting as a kind of cache when you're building new, updating memory and things like that. I'm wondering in this version of, okay, the amygdala is not just a passive pass-through, but instead it's doing learning. It has sparse connectivity, just like you see sparse connectivity in CA3, as opposed to what you might find in sensory cortex, neocortex. Do you know of papers and experiments showing fear memory becoming independent of amygdala, the same way that, contextual memory becomes independent of hippocampus?
It's a great question.
Looks like Ariel might have an answer to that. He's got his hand up.
Yeah, I'm going to throw that to anybody else that knows.
I do not have an answer. I have a follow-up to Randall's. I'll give anyone else a moment to jump in if they have an answer. No,
I should know this because I read Ladeau's book on the synaptic self, and I'm sure that he talked about this. There are a lot of fear memory systems, and the frontal cortex has its own fear memory. So there is at the very least there is a cortical other side that can be dissociated.
But what you don't know is whether it becomes offloaded to another site from amygdala after a certain period of time.
That's right. I don't know.
Yeah, I'm basically curious about the same stuff. So like with non-necessarily fear memories, other memories that have been recorded, there's been some cool work where you see like simultaneous formation of engrams in the hippocampus, but also in cortex. And slowly what you see over time is for like long-term recall. So like days to weeks later, the cortex becomes more important. The hippocampus starts to wipe the original memory. I'm curious whether the same thing happens for the amygdala or a different thing happens, where like do those lateral amygdala neurons store the memory forever or just temporarily until it becomes cortex dependent? And like it's not part of this paper, but I'm curious if anyone's seen whether they've done studies trying to see if those lateral amygdala neurons are still important weeks later.
Well, I know one thing that they've done is they've done a bunch of studies on, I forget what they call it, but when you give the same stimulus but without a shock multiple times, and you essentially, the animal learns to not freeze. They've done studies to ask where that is, and if you've forgotten a memory that way, there's a technical term for it, but I'm blanking on it right now. If you've forgotten a memory that way, then if you get one more shock with a tone, with the original tone, then that memory comes back much more quickly than you would think. So these are definitely extended periods of time. And the research that I've seen on that is all also based in the amygdala. And they make an argument that the amygdala is actually learning not to forget the original memory, but to learn a new memory that cancels out the original memory.
Interesting.
Yeah.
Another thing
called
that's
extinction,
sort of in
by
the
the way.
same.
Thank you.
Another thing that's in kind of along the same lines, where I'm wondering about, how much of this is happening in amygdala and what amygdala can be responsible for, is I'm wondering about any tests involving the capacity and also the ability to distinguish, like how many different kinds of fear memories can one distinguish and how many fear memories can one learn in what span of time? Like, can you learn 1,000 different fear memories that you can distinguish in one day, for instance, or something like that?
Yeah, I have no idea, and it's interesting that this paper in the abstract makes it seem like there's a whole bunch of memories, but they never unpack that to say how many. I have no idea. It's a good question, and given that there's 10 to 30% of neurons in an engram, that's the kind of thing that you could potentially calculate and then test. All right, so if there's no more pressing questions, I wanted to throw this at you, and then we can have a back and forth on this, if that's okay. Any other? Nope. Okay. Okay, so I propose a memory decoding experiment, okay, based upon this. Here we go. I'm going to have some diagrams later, but I'm going to give most of it in text right now. So create two distinct optogenetic memories, like the Nabavi paper that I just showed. one would be with one channel rhodopsin that has one excitation frequency, and crimson R which is another optogenetic channel that has a different excitation frequency. So you could basically have two fake tones, and the axons associated with those two fake tones one could be labeled in green fluorescent protein, the other could be labeled in a red fluorescent protein. This could also potentially be replaced with optogenetic tagging with immediate early gene type stuff, but I don't know if that would work or not. So the other thing, same animal, would undergo immediate early gene labeling of the engram cells associated with both of those fear memories. So two fear memories, two different tones, two different fake tones, right? This paper that we covered a while back, Abdu 2018, showed that you could label two separate lateral amygdala engrams, one labeled using CFOS TTA system and the other using CFOS antibody labels after and then sacrifice the animal. Okay, that puts a bunch of fluorescence in that labels the engram cells and the auditory cortex neurons. Then you take a subset, a relatively small subset, maybe a couple hundred microns on a side, electron microscopy volume that allows you to get all of the connections within the lateral amygdala that you need. And then you attempt to decode the memory, and I'll show you how to do this, based upon only that EM connectivity. And you do this by sorting and stuff. I'll show you that. And then you compare that to the ground truth of those labels. And so this is a diagram that kind of diagrammatically shows what I was talking about. So let's see, you've got these two labeled fake auditory signals that are associated with, the green is associated with the yellow engram cells, and the red is associated with the blue engram cells. And then you do an electron microscopy volume that incorporates, let's say, all of those connections. And you would get something like this. So this would be if you took all the lateral amygdala to lateral amygdala connections that you get in this electron microscopy diagram, you get some connection matrix like this. But the idea is that you could sort that. And if you sorted that, there would be clusters of highly interconnected ones. That's what this paper is really, really saying. It's saying that, hey, if you sort the connectome of the lateral amygdala, you will see engram clusters. And so you sort the neurons based upon those. And then this is showing the connections from the auditory cortex axons to those neurons that are sorted. And so what I hope you see is you see stripes that are kind of saying, hey, this axon was active for this memory, but this axon wasn't. If you thresholded those, you should get something like this. And so this would be the predicted memory, the predicted sound, right, for engram 1. And this would be the predicted sound for engram 2. Okay, now that's all based on electron microscopy so far.
Can I ask a quick question?
Yes, please.
So this looks super familiar somehow, and I'm trying to remember if it's familiar to me from modeling or from experiment. Have you checked to see if there are experiments quite like this, perhaps in hippocampus?
I mean, I was kind of hoping that if something like this would pop out of your of the paper that you're doing next time, apparently.
Oh, okay. Well, then I can chime in on that, because I mean, it sounds like a good plan, and it aligns with a thing that I was thinking as I was going through the paper. I was thinking, okay, wonderful experiments. What I would love to see is I would love to see some high throughput images of all of this, just to actually see something in the circuitry. But Yeah, so this is going definitely in the right direction.
This is a pure electrophysiology paper. Yeah.
Yeah, you want to learn the circuits a bit, so definitely. But it's just that this sorting and then seeing how your input connections are being activated along with that, and all of this, it just looks like I've seen this in hippocampal paper somewhere, and I just don't remember if that was modeling or experiment.
We should definitely look that up. Yeah, I would love. I haven't seen this. I've seen. Sebastian Sung had a decoding memory kind of theoretical paper once that had diagrams like this. That's one place where I've seen it.
Okay.
In any case, the idea is that all of that prediction was based only on the electron microscopy. But if we were to overlay the fluorescence, this is what you should get. All this is hypothetical, right? I mean, it's this would be beautiful if it happened this way, right? But the idea is that the engram, the lateral amygdala engram number 1 should be should have the yellow immediate early gene labels in it, right? And it should be preferentially connected to the green fluorescently labeled axons. That there are ways to get that information into the electron microscopy. So this is a correlated electron microscopy and light microscopy study. But in any case, the idea is that you would literally have the ground truth, and so the predicted vectors, obviously they won't be exactly the same as the ground truth vectors, but you could compare the two, and you could compare the two based on, let's say, the dot product or something, right? And compare it to a bunch of random vectors that you chose. And if only one out of, let's say, 2 to the 10 random vectors were as close to the ground truth memory vector as the decoded predicted vector that we got, then we could say that 10 bits of information was successfully decoded. And so to end this up, because this is the end, I propose to the group that this would meet some kind of qualification for being a non-trivial memory decoding, as long as the number of bits of information decoded was itself non-trivial, let's say 10 bits. And that's it.
Thanks. Awesome presentation, and also awesome proposed experiment. I guess we can debate whether it's non-trivial. You already had mentioned to me earlier that you thought some of us might think it's potentially trivial. And I think it might be, in the sense that, say, the kind of information you could pick up in an MRI to distinguish in, Jack Gallant's kinds of experiments between whether someone is seeing one image or another will also have a certain number of bits, and they may not overlap by much. And then you could say we're really is just distinguishing two things. But still, I like your approach better because of the random vector part of it. And I could imagine that, you can easily take this from having 2 engrams that you're comparing to 3 or 4, if there were enough ways to color things, I guess. That's the question.
There's one paper that I saw that does 3 labels for these immediate early gene stuff, but that's pushing the very limit. Most papers just do 1, and they're very difficult at that.
Yeah.
A question, a slightly technical question that might just reveal my computer science, information theory ignorance, but it's 10 bits because you're comparing like how many let you know which of the 2 engrams it's from versus how many neurons are just corresponding to like completely disconnected random other neurons. If you increase the population of neurons you were sampling from, does that mean that the bit number would go up, or what's the ultimate determinant of how many bits?
Actually, I was I was asking some people around here if that is a proper way of thinking about information. I don't know if this is the proper way of thinking about information. I'm thinking of it from the point of view of I have a lack of information, and I'm narrowing down between possibilities. And if I narrow it down, like 1 out of 1,000, then that should be 10 bits. But
yeah, the bit measure is typically a question of how many different things can you distinguish. So, for example, that's why I say, if we have a computer byte with 8 bits in it, you can distinguish 256 different things, and that's 8 bits of information.
Yeah,
but yeah, I mean, this is a very crucial point, right? Because it's kind of like, I think this gets to the heart of what non-trivial would be. So go ahead, Aro.
Yeah, I was just trying to clarify whether there's like 3 possibilities. It's like engram 1, engram 2, or like random neuron, or whether we're considering it's like more possibilities that we're detecting out of.
I mean, essentially, this is if you were thinking about this as sounds, right? If you knew, which we don't, okay, I tried to think of an experiment that could do this, and I couldn't really. If you somehow knew the exact receptive field properties, frequency tunings, let's say, of the auditory cortex neurons, and there were 1,000 of them, and that palette was palette. Anyway, that set of neurons could give all manner of different sounds, right? Then if you said, I've looked at this connectome and I know exactly which sound you were playing when the mouse was shocked, right? And it was Beethoven's Fifth or something, right? You played Beethoven's Fifth for this mouse when you shocked it. Right out of like A Clockwork Orange or something. And that would be more than just saying there was a memory or not. That would be a non-trivial decode, right?
Yeah.
That would be non-trivial. That's just like the bird song experiment in a sense, right? Trying to determine different songs. Exactly. Why did you have to mention Beethoven's Fifth? Because now I'm thinking temporal, and now I'm thinking, so what evidence do we have in amygdala that it can do heteroassociative things and distinguish, temporally extended things rather than a single tone and all of that?
It's a good question. I mean, one thing is this is not the hippocampus, right? It's
right.
This is I think it's an older structure, whatever that means. Maybe not. But it's got similarity to striatum.
But remember the last paper, right? So the context remembered by hippocampus, which can be a sequence, gets associated with something in amygdala, gets basically one label or something like that, or at least that's the theory then.
Absolutely, but I would argue that without any evidence whatsoever, is that we have areas in the brain that are that turn temporal information into a flat representation. The auditory cortex does this, right? The hippocampus does this, perhaps. Okay.
For sequences of whatever, yeah.
Yeah, and so the amygdala doesn't have to do that. It could be relying upon a flattening of a temporal sequence that is happening in the auditory cortex, and that way it could be sensitive to a temporal sequence, but it doesn't have to do the computation itself.
Mm-hmm. Yeah.
That having said that, I don't know. I mean, it's like if you were talking about the hippocampus, you would expect not just like a single engram. This is why I think the amygdala is simpler than the hippocampus.
Yeah, and I think that doesn't really matter. I think I was just distracted by the thought about temporal whatever, but since we just assumed that whatever it is that amygdala depends on beyond that, coding in sensory cortex, coding in hippocampus, whatever, as long as you have X distinguishable things, then you can strive for an experiment where you do something non-trivial by being able to identify a specific memory out of a set of possible memories. Yeah.
Wasn't there a previous, maybe it was just an in vivo paper where they did a distinguishing high versus low auditory tone in the auditory cortex that has some analogies to what you're describing here?
So there were a couple of papers that we did. One is this Abdu paper that I referenced, but I think the one you're talking about is Tony Zadar, his group, that was distinguishing a high and a low tone going into the striatum, and they were saying, did the mouse learn or the rat learn to lick right or left?
Yeah, that was the one.
High tone or low tone. Yes, and they marked. So it's a good point. They used, I believe, they could have done two ways. I don't remember how they did. They could have injected the auditory cortex. The auditory cortex has a bit of a tonotopy. So they could have injected to the high frequency part of the auditory cortex. But I don't think that's what they did. I think they used some kind of immediate early gene labeling to have optogenetics be expressed only in those auditory cortex cells that were active for a particular tone that they played the animal. I'd have to review the paper.
Yeah. I mean, insofar as you have an experimental setup where, as like Randall was saying, like how many like distinct tone like associations can you teach the rodents? But I certainly feel, I mean, with 2, maybe it's already enough to be non-trivial. But if you're getting to like 5 or 10 or 20 or something, like I think it'd be very hard to argue that if you were pulling the memories out, that it was still non-trivial. I mean, for me, the key things are like, is it distinct to like this animal? Is it like something from its learned experience as opposed to like a generic evolutionary style instinctual memory? And how like specific is it? And like arbitrary is it? Like it's that it's like paired this arbitrary sound with this arbitrary experience. I mean, it seems like a reasonable candidate to me. Who would argue this is still trivial?
I wouldn't argue that. As in the context of the whole memory decoding challenge, since you're also trying to get people to express whether or not they are satisfied with the idea that engrams are represented by synaptic strength or connection strength expressed, for example, through LTP, those people who have different ideas about what else is required to encode a memory, they're not necessarily get from this particular proposal what they're looking for. But I wouldn't know how to stuff that in there as well, because you can't test everything. And also you don't know what other recommendations they would have.
Yeah. Andy, what do you think? You think this would be trivial?
No.
No?
No, I think it'd be non-trivial.
Yeah. The other reason why I was I think anything that we gave a non-trivial moniker to would have to say something about it. It's like if you're given a connectome, where do you start to get a handle on in order to get information? And the idea is that this experiment is saying, look, you need to look for the engram ensembles. If there wasn't a connections between those that you could pull out of the EM connectome, then you wouldn't have any place to start. You wouldn't be able to distinguish these two, and so it leads to other experiments. I think this is what you guys are saying, that how many of these ensembles could you pick out reliably from any particular area, including like the hippocampus or something, where these the same type of principle should be at work.
Yeah, and there's another level, of course, that I'm interested in when it comes to memory decoding, because I always think of it as, say, a whole pipeline of encoding and decoding And then you're taking a snapshot here right at the amygdala, and you're looking at engrams in there. Now, if those engrams themselves are not sufficient to describe exactly what is encoded there, they're only able to distinguish between different things that are encoded. That still leaves us with, looking forward to brain preservation and whole brain emulation and all that, leaves us still with the question of, if you had a slab of brain and you want to reconstruct something in there and get back, what did this person learn? What is in there? What did this animal learn? Being able to distinguish isn't enough for that. At some point you have to get to mapping out that whole pipeline. So all these connections, so that you can then represent, oh yes, so this, or at least it has to work in the context. Even if you can't tell what is in someone's memory, that they heard Beethoven's Fifth, right? Because it's not just distinguishing between things, but, oh, this is Beethoven's Fifth. Then even if you can't figure out how to do that, at least as a whole, it should work together. It should, if you then ran the entire pipeline, it should function. It should be able to recognize Beethoven's Fifth, right? Because just the slice of amygdala can't do that, obviously. Right. So I know I'm just rambling now about like thoughts on my mind, but it's a great experiment anyway.
Okay. And I offered up as just an example, because I think, and we're over time, so this is my attempt at a wrap up. We really should be talking about the memory decoding challenge and getting a bunch of ideas on what would eventually win something like that. And so I offer this up as a concrete example that is not may not have all the stuff that we're looking for, but it's kind of getting in that direction. And so so with that, any other points?
What I would add to that is I like that you have there an example that tells people concretely what you're looking for in the memory decoding challenge. And then what would be nice if if we distribute this, if you show this around, if Ariel puts it out there, is if people come back with a list of things they think are missing or that one would still need to know, or critiques, etc., so that you can kind of build up more that explains, okay, all of these things fit into the memory decoding challenge, kind of almost what falls within, what falls without, kind of, lists.
Yeah. All right. Thanks everybody.
Thanks all of you. Thanks for
you.
the presentation, Ken.
Thanks.
Thank you, Ken.
Thanks everyone.